Welcome to microbiology class. In this video, we are going to see methods used to demonstrate the motility of microorganisms. Motility. Motility means movement. The movement of microorganisms. See, the movement is one criteria which is used for identification of microorganisms. Like a person moving. You may be remembering the film stars, their style of movement. Based on their style of movement, without seeing their face, you can identify them. For example, Rajnikant's style of walking. Without seeing his face itself, you will be able to identify by the way he moves. So like that, the movement, the type of movement or motility is used to identify microorganisms. There are various methods are available. Before that, we will see for movement of human beings and other animals, we have legs. That is a locomotory organ for us. Locomotion. For movement, we have legs. What about microorganisms? There are motile organisms are also there. Non-motile organisms are also there. The motile organisms are moved from one place to another. Okay, they can move from one place to another with the help of locomotory organs. There are various locomotory organs present in microorganisms. Some will have flagellum, some will have cilia, some will have pseudopodia. Okay, so these are the common structures seen for locomotion. Cilia, flagellum, and pseudopodia. Protozoan microorganism, protozoa, parasites like amoeba, Giardia intestinalis, Leishmania, Balantidium coli, Paramecium. So that type of organisms are having these locomotory organs. For example, pseudopodia is seen in amoeba, entamoeba and other amoeba. We can see a structure known as pseudopodium. Giardia intestinalis, that type of organisms will have a locomotory organ called flagellum. There is another locomotory structure, it is known as cilia. Okay, Balantidium coli, Paramecium, they are having this type of locomotory organ. Flagellum is a common locomotory organ present in bacteria. There are differences between bacterial flagellum and this protozoan flagellum. But as far as bacteria are concerned, the locomotory structure is only flagellum. Okay, only flagellum. Some bacteria may have some other projections known as fimbriae or pilae. They are not locomotory organs. They are only used for attachment. The locomotory organ structure present in bacteria is flagellum. Some bacteria will have only one flagellum, some will have more than one. The type of arrangement also will be different. Some all over the body you can see flagellum, some will have only in poles there will be flagellum will be present. So like that, different types of arrangement of flagellum can be seen in bacteria. So based on this arrangement of flagellum and based on the number of the flagellum, the type of movement, the speed of the movement of the organism also will be different. Some, if there are more flagellum means the bacteria can move faster. If flagellum is present all over the body, the bacteria can move in any direction. If flagellum is only present only in one pole, in one end, it can move only in one direction. Okay, so based on the type of arrangement of the flagellum, the motility also will be different. Sometimes we known as a rocket-like movement. Okay, darting motility, tumbling motility, corkscrew type of motility, okay, falling leaf type of motility. So there are different types of motility you can see. Based on this motility, you can identify the organism. Now, how can you identify whether the organism is motile or not? There are motile organisms are also there, non-motile organisms are also there. For example, your bacteria, among bacteria, Staphylococcus, most of the cocci are non-motile. Staphylococcus, Streptococcus, they are non-motile. But other bacteria like Escherichia coli, Salmonella, those organisms, Vibrio cholerae, they are motile. If you see the bacilli, rod-shaped bacteria, the Klebsiella are non-motile, Shigella are non-motile, but Salmonella, Escherichia coli, Vibrio cholerae, Pseudomonas, those Proteus, those type of organisms are motile. So this motility is one criteria used for identification of bacteria and other microorganisms. Not only microorganism, you can see the motility of sperms. Okay, sperm motility, you can see other water larval forms. Those motility also can be observed under microscope. There are various methods available for demonstration of motility. Microscopic methods are also available. Non-microscopic methods are also available. Microscopic methods, one common method is a wet mount preparation. Already we have explained about wet mount preparation. Wet mount preparation. You take a slide, put a drop of the culture or growth of or sample, then put a cover slip, observe under microscope. You can see the motility of the organism. So that is wet mount preparation. Next one is hanging drop preparation. Hanging drop preparation. For that you need a cavity slide, and in that cavity slide, using the cavity slide, we can do a preparation. It is called hanging drop preparation. That is another microscopic method. Then another method which is used to see the motility of anaerobic bacteria. Anaerobic bacteria. Anaerobic bacteria means bacteria which cannot live in the presence of oxygen. On exposure to oxygen, they will die. So by doing this wet mount and hanging drop preparation, you will not be able to see the motility of anaerobic bacteria. To see their motility, we use one time method known as capillary tube method. Capillary tube method. The culture of anaerobic bacteria will be taken in a capillary tube. Both ends will be closed, and that capillary tube will be observed under microscope. So that is for anaerobic bacteria. Then there are non-microscopic methods like using semi-solid agar culture methods. There are some special mediums are available like mannitol motility medium. We will inoculate the bacterial culture into that by a stab culture and incubate it. After incubation, you can see the spreading type of colonies. The growth will be swarming growth, you can see. If that type of swarming growth is seen, that means the bacteria are motile. So that is a culture method. Okay, and these are the methods used for demonstration of motility. Wet mount preparation, hanging drop preparation, then capillary tube method for anaerobic bacteria, and culture methods using semi-solid agars like mannitol motility medium. Okay, so in this section, we will be seeing one video about this hanging drop preparation and wet mount preparation, which will be demonstrating the motility of microorganism. Okay, so that's about motility and hanging drop preparation or demonstration of motility. Thank you. Following this, you can see a video and it will show you the demonstration of motility under microscope.
In this short video, you will learn how to perform the motility test. This test is a useful tool to differentiate bacteria by detecting if a bacteria is motile due to the presence of flagella. After this program, you'll be able to discuss the steps to perform both the wet and tube motility tests and identify a positive and negative reaction in motility media and on a wet mount. There are two primary methods for performing a motility test: the wet mount method and the motility media method. There are several species of bacteria that are motile by their attached flagella. The wet mount involves observing this motility in a drop of saline or broth. Bacteria can usually be seen moving at different angles of direction in the solution as compared to bacteria moving as a result of Brownian motion, which is the normal drifting of particles in a solution. Motility media is a semi-solid media which is the result of a low concentration of agar. Because the media is semi-solid, there is an easy visual determination of motility which appears as diffuse growth outward from the line of inoculation or turbidity of the media. If using media with TTC, try trifentrizoleum chloride, which is a colorless salt that turns a red color when becomes reduced as a result of bacterial metabolism, you will see a red color in the line of inoculation and a pink color that diffuses out from it if the organism is motile. For this procedure, you'll need the following: sterile gloves, laboratory coat, and safety glasses, sterile Pasteur pipettes, large 2x3 glass slides, loops and/or needles, cover slips. For this presentation, we'll be using two types of motility media: motility media with and without TTC. Motility media with TTC is a colorless salt that turns a red color when it becomes reduced as a result of bacterial metabolism. For our samples, we have 2 agar plates containing 2 isolated colony types that are approximately 18 hours old. Before you can perform the wet mount to determine motility, it is best if the organism is placed in enrichment broth such as BHI, brain heart infusion, or TSB, tryptase soy broth. Using a plastic loop, isolated colonies are picked from a blood agar plate and transferred into a labeled tube of BHI broth. The loop is then discarded into the sharps container. Using a sterile Pasteur pipette, remove a small amount of broth and place a single drop on the center of a large glass slide. Place a glass cover slip over the sample and allow the organisms to settle for a minute. Next, observe the sample under high power 40x on a microscope. Directional motility is recorded as a positive test. If the direct test is negative, either repeat it with the tube test or incubate the broth at 35 degrees for up to 18 to 24 hours. After 18 to 24 hours, remove the broth from the incubator and place it on your bench. Repeat the steps for observing motility. With the sterile inoculating needle, pick an isolated colony and stab the labeled tube medium straight down through the center to a depth of 1/2 inch from the bottom of the tube. When stabbing the agar with the needle, be sure to keep the needle in the same line it entered, as it is removed from the medium. Incubate at 35 degrees for 18 to 24 hours. A positive test in motility media without TTC is diffuse growth away from the site where the media was inoculated or turbidity of the media. A negative test is indicated where there is only growth along the site of the inoculum and the media is clear around it. For TTC media, a positive test is indicated by a red turbid area that extends away from the line that the medium was inoculated and the media is turbid. If there is red growth only along the line of inoculum and the media is clear around it, then the test is negative. To review, for wet mount, place a drop of the broth containing isolated organism on a slide. Cover with a cover slip. Look for directional movement of bacteria. If negative, repeat after incubation or with a tube test. For motility media with or without TTC. Pick a colony with needle. Stab motility straight down 1/2 inch from the bottom of the tube. After 18 to 24 hours, look for turbidity or red turbid area extending away from the stab in the medium.